Single cell, it's a fundamental unit of biology. It's really important to look at different cell types within the system. But right now, a lot of the existing tools to analyze single cell data are geared towards programmers who know how to manipulate and the underlying data types. And also, all of the tools available are only supporting tens of hundreds of cells. But the motivation behind this project is to come up with a tool that supports thousands of cells and tens of thousands of cells, and also allow scientists without much programming experience to look at the data and try to figure out what are the different cell types in this data set. So for example, what you're seeing here is 3,000 cells that we collected from blood at Next Genomics. And we're going to use this data set to demonstrate the use of our application, which is currently named VASCO. So I let JC explain the origin of this name.
Yeah, I came up with this name. Do you remember your history class, Vasco da Gama? Mm-hmm. Yeah, so he's an explorer, a Portuguese explorer, and I came with this idea because I just came back from a trip in Portugal with my girlfriend. So history geek, but the name of the app is a visualization app for single cell exploration. So that's about it. We will explore some single cell data with you.
So currently we enable two features. The first feature allows you to visualize markers among different cells. And then the second feature allows you to explore different clusters within your cell type. So first, I think we're going to go over the gene visualization part.
So typically, we start with unlabeled clusters, and we want to know what are the questions we may have to ask is, what are these clusters? And one way to get an idea of what these clusters are is to look at the expression of these points. So the expression of these clusters. So what we see here is a scatter plot of the 3,000 cells. The color represents the level of gene expression. The gene up there is LGAL-S1, which is a marker for monocytes. And so we see here that that cluster up there is highlighted pink, which represents the level of expression. So we might infer that this cluster up there are the monocytes. What Ogun just demonstrated is that our tool we can select multiple genes to compare. We can select up to 4 genes, and so you can like change the color scale to, let's say, a maximum of 0.2 to increase the contrast. We can also change the color of here to try to visualize the different populations. And so CD3D is like a marker for CD4 cells, and so we might infer that this is associated with CD4s. Also implemented is a box plot feature, which shows the level expressions of the genes per cluster. And in addition, we can also save these figures.
So once users have identified potential tags or clusters, we want to give them the ability to annotate these tags so that they can do further analysis. So we've implemented the ability to select a cluster and to annotate it. So say we identified CD4 cells in the last tab, we can annotate them as CD4 cells for future analysis. So we also want to allow the user to compare between clusters so they can identify like novel markers or other kind of discriminatory analysis between clusters. So the user has the ability to either select from predefined clusters, either from the clustering algorithm or from their own annotation, and compare gene expression between two clusters. So, Ogun, do you want to talk about it?
Sure. What we get as a result of this is the mean expression of the two groups that we just looked at, and the maximum fold change of the genes. And this can be sorted in any way, and if you really want, you can download the data as well. But since I am selecting a particularly poor cluster right now, the difference is not particularly high. But yeah, this data is downloadable, and if you want, you can, by default, this guy shows you the top 20 higher expressed and under expressed genes between the cluster, but if you want to see an expression of any individual